Proteins, that comes from the Greek word first, that means. So we are going to talk about the proteins. Their function is they part of the defense mechanism in the form of antibodies and antigens. They are part of the communication in the form of hormones and receptors for the cells. They act as biological catalysts like enzymes. They act as a storage for embryos, like in the form of albumin. They help with the transport like hemoglobins for the oxygen and channel proteins for the active transport. They are part in the movement as a actin and myosin, and they also part for the structure to support different parts in the cell like collagens and keratin. Amino acids are going to, many amino acids form polypeptides, and as the polypeptides get folded in the rough endoplasmatic reticulum, they will gain a three-dimensional form, the protein form. This is how the protein, a general protein, look like, with the amino group on the left, and the right is the carboxyl group. The central carbon is called the alpha carbon. That is the alpha carbon. And the R is the side chain that is responsible for the chemical behavior or characteristics of the amino acid. It can be nonpolar, polar, acidic, that is negatively charged, or basic, that is positively charged. We have 20 different amino acids. One amino acid and another amino acid is going to bind together through dehydration reaction. Their amino group will react with the carboxyl, the other one's carboxyl group, and the water is leaving the system, dehydration reaction, and it's going to form a polypeptide. The carboxyl's oxygen group will be a little bit partially negatively charged, and the hydrogen on the amino group will be positively charged a little bit, so the hydrogen bond will form between these two atoms. This will be the secondary characteristics for the protein. So this is a protein, actually a peptide, polypeptide. I highlighted the peptide backbone, polypeptide backbone. And also I think, yeah, this red is going to be the peptide bond between the carboxyl and amino group. That it used to be the carboxyl and amino group. Now, protein structure. The protein has four different structures: the primary structure, secondary, tertiary, and quaternary. The primary structure comes from the order of amino acids in the polypeptide chain. The secondary structure comes from the hydrogen bond within the backbones. Remember, when oxygen is partially negative and the hydrogen is partially positive in the polypeptide, they are going to bind together through hydrogen bonds. They can form alpha helix if we are in one polypeptide. It's going to be a helical structure, and beta pleated sheet will form when between two polypeptides next to each other. The tertiary structure comes from the interconnection between the R groups. Can be weak van der Waals or hydrogen bonds, and ionic bonds also be, or disulfide bridges with the cysteine that contains sulfur atoms. The quaternary structure is happening if we have more than one polypeptide and they bind together. Not all of the polypeptides have quaternary structures. collagen and hemoglobin they have. The folding and the structure is really important, because if misfolding will happen, this is when Alzheimer disease or Parkinson disease will form. The last unit is the nucleic acids, the central dogma that states that DNA is going to be transcribed into messenger RNA in the nucleus, and the messenger RNA will leave the nucleus and will be translated into protein. The nucleic acid, we are going to talk about the DNA and RNA. Nucleic acid is made up of many nucleotides, so we can call it polynucleotide, the nucleic acid. The general structure for the nucleic acid, for the nucleotides, we're at the nucleotides. The pentose is a 5-carbon sugar. The 5-carbon sugar is here. We have to number the carbons in the sugar: first primer, second, third, fourth, and fifth primer. The sugar can be deoxyribose for the DNA, and in this case, the second primer, on the second primer carbon, there's no hydroxyl group. This is really the difference. For the ribose, that is part of the RNA, the second primer carbon contains the hydroxyl group. Both DNA and RNA, or deoxyribose and ribose, on the third carbon contains a hydroxyl group. To the first prime, the nitrogenous base is going to bind to the pentose. The pyrimidine that contains, and the purine, the pyrimidine contains 1 ring, and it's the cytosine, thymine, and uracil. The purine contains 2 rings, and the adenine and guanine they are the purine bases. If they usually pick up hydrogen ions, so they act as a base, hence comes the nitrogenous base come from. To the 5th prime carbon, the phosphate group binds, and what else? Okay, so nucleotides. Nucleotides I'm going to just draw around with a red pencil. Contains the phosphate group, the pentose, and the nitrogenous base. With the blue pencil, I'm going to draw around the pentose and the nitrogenous base without the phosphate group, and they are called as nucleosides. Nucleoside without phosphorus, without phosphate group. The DNA and RNA, they run anti-parallel to each other. We will discuss that in class. And they are complementary strands to each other. And if you remember that from junior year, this is very important. And that's it for the proteins and DNAs.